Right. Well, it's a pleasure to be here and talk to you all today. And the topic's a little different than what's been covered so far. What I would like to talk about is essentially present and future production of PET radiopharmaceuticals, regulatory requirements, and clinical use. Okay. Not quite sure how this guy works. Okay. How do I forward this? That's one question. Excuse me?
Right here, the green button's forward and the red button's back.
Okay, and then this is also a pointer? Yeah. Got it. Okay. What I was asked to talk about is kind of an overview, basics of the cyclotron, production of PET radiopharmaceuticals, a little bit of historical overview of the PET regulatory and FDA approval process, clinical PET drugs, investigational PET radiopharmaceuticals, and then some future vision that I have. So it's a lot to cover in a short amount of time, but I'll do my best. So essentially, what a cyclotron is the acceleration. It's a device that accelerates particles in a circular path. They're actually maintained in between the poles of the electromagnet. So what's going to happen is the particle will be injected essentially into the center of that, the Ds, and then it will be essentially traveling to the outer area of the cyclotron where then it's ejected and it hits its target material. So depending on the target material that you have, you make various kinds of radionuclides. As far as PET radiopharmaceuticals, there are a number of common ones that we use. They're not a full listing, but we make all of these on our cyclotron routinely. O18, and essentially these are the nuclear reactions for the production of these isotopes. So we start with stable O18 to make fluorine-18, which is the most common, excuse me, went backwards here, a little too fast, most commonly used PET nuclide. We also make carbon-11, have a 20-minute half-life, N13 a 10-minute, and oxygen-15 a 2-minute half-life. So within those half-lives, you can imagine that it's difficult to produce, synthesize, do quality control, and inject it in the final product in a human. I mean, it really is a significant undertaking. What I meant in terms of the targets that we use gases, we use liquids, we also use metals. So in terms of the production of water, O15 water and oxygen, we make O15 oxygen, and we can either use regular nitrogen, which is a cheaper approach, but we have to have deuterons on our cyclotron to be able to do it. We can use stable nitrogen, but then we can do a PN reaction, so that's just a more expensive method. We happen to have 2 cyclotrons that we do have deuterons, so we're fortunate we can use the water target for our production of N13. We have also on our one cyclotron used the enriched material, and again, it's just a more expensive process. Carbon-11, we make a number of things. C11 acetate is an agent that we were using prior to essentially the approval of C11 choline by Mayo Clinic. I'll talk a bit about that. We also make fluorodopa. It's another non-approved research tracer. We start with O18 gas, and then we end up with F18 or F2. Again, it's a different target material. This is the liquid that we end up starting to make fluorine. We use liquid to produce the F2 gas to make the fluorodopa, which is an electrophilic method currently. We're switching it over to nucleophilic, but again, we used a different target. Liquid targets, O18 for fluorine, O16 for N13. Metal targets. We also produce a couple of longer-lived PET isotopes that we ship across the United States. We produce copper-64, which again has about 2.8-day half-life. And again, we start with nickel. What we do is we take this enriched nickel-64, we plate it on a gold disc, we irradiate it with protons, and we produce the copper-64. And we do have a number of research radiopharmaceuticals that we produce at Washington University, and across the United States there are a number as well. This is just a picture of our most recently installed cyclotron. We actually have 4 cyclotrons. We have 2 older machines, which are positive ion machines. In other words, we accelerate the protons or the deuterons. Actually, we're both down to just accelerating protons. But why is that a disadvantage? It's essentially we activate the machine. It's a much higher risk because you have essentially a radioactive machine once you finish bombarding for these various nuclides. And so they have to be kind of away from everyone. Our positive ion machines are actually in the basement below our basement. So they're certainly kept away. And the newer generation cyclotrons are negative ion machines, so we ionize hydrogen gas typically, or deuterium gas, depending on what we want to produce, and then we accelerate the negative ion. And that does not interact with the machine as much, and so again, it's a much safer machine. You can turn it off, and essentially, very shortly afterwards, you can walk right up next to the machine. There certainly will be, a dose rate from the machine, but it's not like your positive ion machine that you can't be near. And as far as, excuse me, this little thing, sorry, I'll get this right yet. Okay, so what we make in terms of yields, this is the standard isotope production of the TR19. It's variable energy and it runs from 14 to 19 MeV. In other words, we can change that energy, and we accelerate deuterons from 8 to 9 MeV beam. But when we're making our traditional fluorine, oxygen, nitrogen, we're doing them in our regular cyclotron, which is here. They bombard targets that are at the end of the range. But now with our solid targets, this is the extended beam line. And again, when we're running our targets, this is not, again, shielded. So we're in a vault that, again, is in our basement. We don't go near that again. So there's other radiation considerations when you're using solid target irradiations. Some, like the GE machine, they sometimes build vaults. They're essentially concrete areas that you can then walk closer to the machine. We make, again, on this machine, we're able to produce about 3.2 curies in 30 minutes of the carbon-11. For fluorine, we can make up to about 10 curies in a couple of hours. And N13, we really haven't used it to do the production of our N13. That's still over on the RDS. It's only been installed about 1.5 years now, so we're still bringing things over. The copper-64 productions come over. Zirconium-89 is another nuclide that is being used investigationally, and that's one that we currently make on one of our positive ion machines, and we're in the process of making the target so we can move it over to our new area. This is just an internal look at the Ds of the cyclotron. If you've seen the inside of a GE machine, essentially this is what you see. They're vertical. That was what I showed you or pointed out for our traditional. And what happens with the Ds essentially is that when the particle is injected into the center, these Ds change charge, positive to negative, about 10 to the 6th time per second. So again, what we're trying to do is attract that negative ion to the positively charged D, and we do it and we maintain it in a circular path. By the end of the path, the particle will have acquired the energy that we are trying to achieve. Then it will essentially, the particle then moves through a stripping foil. Carbon stripping foil pulls off the electron, and we're left with the proton. The proton then irradiates our target. So again, we have the same nuclear reaction. It's just that the acceleration is more preferred. This is our older. It was installed in 2001. It's a Siemens RDS Eclipse. And this machine, again, these are the self-shields. And what I've tried to picture here, too, is, again, this machine is a horizontal machine. It's about 5.5 feet in diameter. So again, the same acceleration occurs, just in a different plane. And the shields are always closed when we're operating the machine. These machines also run under high vacuum. So again, if we need to work on the machines to replace or rebuild targets, we have to break the vacuum. And again, once we finish working, we have to pull the vacuum down, and depending on the machine, it can take a significant amount of time. So here you can see then with the machine and the Eclipse shields open. This is just for you to see what these ion sources look like. Again, the machine now is open, obviously. So this is where the gas is actually ionized, and then there's a slit opening in this ion source when it's dropped down in between the Ds, and the machine is closed. So that slit opening is where the particle then is pulled in to be accelerated by the Ds. You can see a better look at the Ds here. Again, they're up on top and down below, so you've got 4 Ds, and again, alternating charge in the ion source. This is just a diagram of what's actually happening in terms of our machine. What's happening as the particle is again moving out from the center, gaining its energy, it then is going to transfer through this stripping foil, and it'll be able to irradiate. A partial beam is stripped, and the other beam can be stripped at target B. So we have 2 different targets. Similarly, For fluorine, for carbon, again, when we want our maximum yields, we bombard 2 targets at the same time, which is very possible. And again, electrons being passed through, stripping, and ending up with your proton. This is just a picture of our schematic of our target. This is our F-18 target. And so again, what you're seeing, this is actually the end that would be bombarded. And this is essentially screwed into the machine at the bottom. So let's move forward here. This is the size of the target. This is an F-18. This is a titanium target. So that's a person's hand that's holding that target. It's not very large. And again, we fill this to a certain point. Fill volume filled from the bottom. We then push pressure on top. We increase the pressure to about 450 psi. We get a much higher yield if we run under pressure. And I just wanted kind of to step through the chemistry process. I know FDG is an old traditional drug, but I just thought I'd show you in terms of, these are just 2 modules that we have at Washington University. So again, what this is this is the GE Fast Lab, and we purchased cassettes. And these have all the chemicals that are needed for the actual chemistry to produce the FDG. It comes to us in a package that's sterilized and pyrogen-free with a single lot number on it for that entire package, which is wonderful. When I started in this field, we made up all those reagents individually ourselves. We made them up in an aseptic manner, but they weren't sterile. We didn't test them to assure they were sterile. We assumed that they were, but it wasn't really important because our final product is sterile filtered, and that's the bottom line. But when we are able to purchase these cassettes, it saves us a lot of time, and we have a sterile product to start, which is great. This is also a GE FXM. Previously, it was a coincidence module. We actually brought this one in-house in 1999, and it's still a workhorse. It's done very well for us. And GE bought the rights of this particular module. And again, we alternate these typically in a day. On the first Monday and Tuesday, we may only make 1 batch, sometimes then, depending on patient load and how late in the day we have to go. Because a 4 o'clock dose can make us have to have 500 millicuries or 700 millicuries at 7 o'clock in the morning when we let the batch out. So it really requires, the later they go, we can usually cover it if they're up to 3 o'clock in the afternoon, otherwise we have to start making another run. This is just a FDG image, which I'm sure you've seen. Just essentially what I wanted to show on this too is the clearance, the ureters and the bladder in terms of the background activity that exists in a normal biodistribution of a patient. And that'll be something I'll show you again as I look at acetate and choline. So then once we finish producing this, we bombard for 2 hours, we bring the activity out of the target, we then run through shielded lines under the floor up to our hot cells. All of this is done in hot cells because obviously the amount of activity that has to be handled can't be handled manually unless you're using remote arms. But all of our modules run again. And then once we finish the product, actually we start the product process by setting up our final product vial in a manner that we insert vent needle and sterile filter needle in that sterile vial, and we have to do it in a laminar flow hood because this is an aseptic process. Once we finish the product, we call it sterile, but sterilization testing takes 2 weeks. So we don't have 2 weeks to test this product with a 110-minute half-life. So we have a lot of things that we have to do to validate our process, to be able to say what we do to the FDA is acceptable and that we do have a sterile product. So we do 3 validation runs, we do complete testing and finalize our sterility before we can move forward and actually make patient batches. So again, these are the tests. Once this is finished, every single day that we make a batch of FDG, we do all of these tests. Again, it takes typically about 20 minutes to do the quality control. So we're looking at, the identity of the nuclide. We're looking at the radiochemical purity, pH, radiochemical and chemical identity. So again, residual solvents. We use solvents that you can't just inject in humans. Acetonitrile is toxic unless we're at a certain level. It's acceptable. So all of this has to be tested. In our institution, residents come in and just observe the process for the understanding of what it takes to get this product to you, because a lot of times things fail. and we're dealing with machines from the opening of our day at 3 a.m. until we deliver final product at 7. That's just one, but a lot can go wrong. And all of our QC instrumentation as well. We deal with high pressure liquid chromatography, gas chromatography. So again, it's really quite an undertaking to accomplish. So with that said, I'm going to step back. PET drugs have been under the FDA regulation since 2012. So this isn't very long ago that we've been dealing with the FDA and the regulatory requirements that have impacted us. FDG started in the academic settings, being made at universities, being prepared and used for clinical studies. But then as insurance companies reimbursed the product, then the FDA became very interested to be involved to assure that we were doing the right thing. So it took them 15 years to give us that regulation. We started in 1995. Actually, it says here, we started meeting with FDA. They were considering it in 95. And then in 1997, the Food, Drug, and Modernization Act required that special good manufacturing practices be written for PET drug production, different than therapeutic drugs. And why would that be? Well, we can't do all the testing on drugs that they can do on sterile, stable compounds. And our half-lives essentially mean we have a product today but none tomorrow. It doesn't sit on the shelf. So again, we're doing all of this testing for every batch. And for a therapeutic compound, they do spot testing. You know, if they make out 1,000 vials of a sterile compound, they'll pull them from the lot and they spot test. They don't test every single vial that goes out into, for you to receive, to be able to make any of the pharmaceuticals, the technetium, or some of the spec things that you're using. So again, this is the argument that we had to put forward because it was impossible. Therapeutic drugs, they might want, say, 10 injections of a drug after you make it before you could release it. Well, each one of those injections on HPLC or a GC takes maybe 15, 20 minutes, so we can't do it. So we spent a lot of time getting this regulation, working with the community and with the FDA. So again, prior to the adoption, though, of this GMP rule, so during those years, we were required to follow our United States Pharmacopoeia, which is our national drug compendia, and it told us the specifications that we had to meet and the process. So again, we had to produce it until we had good manufacturing practice. So as I mentioned, the regulation actually became effective in 2012, and this regulation only applies to PET drugs for routine clinical use. What we have to do if we're interested to make these in our institution is file either a new drug, that would be like for a therapeutic manufacturer if they wanted to, this is IP, or for a brand new PET drug that's never been in a person. For example, fluorodopa, it's been in the academic community for a long time, but in order to have it approved by the FDA, they have to file, the first one has to file the NDA, new drug application. From there on, they can follow the formula that was already on file, and then we can file generic drug applications, which are much easier to file, not so much. The FDA did actually allow us to assume, because of testing, they'd actually done a lot of literature search. FDG had been around for so long, and F18 sodium fluoride and N13 pneumonia all had been in the community. And so these were considered to be safe. That was incredible, so that we didn't have to do all the clinical work, the studies, again, that would be part of the NDA applications. So the FDA helped us out in that regard. They also told us what we have to do to prepare research compounds as well. And that's written into the same law. And it says that, again, we can either follow Part 212, which is harder than USP Chapter 823. So certainly always my recommendation to anyone who's making investigational drugs is to work under USP 823. It's just less paperwork. It also requires that you validate your product, but your instruments themselves, like GCs and HPLCs, don't have to be fully validated. And that's a huge piece, maybe a 150-page document to get a HPLC qualified. So again, what is current good manufacturing practice? This is a term I'm sure you've heard and thrown out. Essentially, it's a rule or it's a regulation that actually the manufacturer has to follow, and it is enforceable in the courts. So that if we do something that is not, that we essentially don't follow this, and they come in and inspect us, then we are liable for that regulation. CGMP is a minimum standard, though. You can always do more, and the FDA is happy to have you do more. But the minimum standard is what is written in the rule. Also, regulations, if you ever read a lot of legal documents, it's not very specific. It tells you you have to do this. It doesn't say how you have to do it. So you have to determine how you want to do that. You have to write standard operating procedures, and you have to follow them. So really, that's the contract that you make. You write the SOP, and then you have to follow it. And it's not acceptable if you don't. So the big thing about the rule is sometimes people, I have experience say, well, we could just end test this product and we could be assured that we have a product that's safe for humans. But that's not what GMP is about. GMP is it's a process. It really is a quality process. So you have to define everything before you do it in writing. All of your operating procedures, your facility procedures, personnel qualification. We have to establish all of this in writing, and then we have to follow it. So we have to then, when we're making the drug, we have to follow the procedures that and in-process controls that we have said we will follow. We have to do all of the quality control, again, written direction procedures. So overall, it's a quality assurance process that is entailed in good manufacturing practice. This just is an example of our facility. We have, again, for producing these PET drugs, you're not required to wear bunny suits. What you are required to do is wear like head covers, face masks, clean laboratory clothing. But you can notice he's got jeans on. He does have booties, and those aren't for radiation safety purposes, but for keeping the area clean. So we use it all the time. This is our laminar flow hood. Excuse me. This is our laminar flow hood. It's an ISO 5 laminar flow. It's the air flows from the top down and then out. So essentially, it's not blowing out into your face. And again, this would be the same type of hood that you'd be making chemotherapy in. It's one, they're in the IV room in a hospital. They have a vertical flow that comes from the back of the hood and blows out into the preparers' faces. So if they're making up really, IV admixtures, they're using vertical flow. But we have to, they're using horizontal, and we use vertical. Sorry. But one other thing, this is our quality control lab. And you're actually looking into this room through a glass window. There's another one of those laminar flow hoods in there where we do our sterility testing. And they want us to keep that area where we're doing sterility testing essentially restricted so that we don't have people walking near these flow hoods to essentially compromise the air quality. So again, this is an ISO 5 area. Our room is probably running at ISO 7 or not ISO, ISO 8, which is what, if you work in traditional nuclear medicine, generators can be eluted in a room that's ISO 8. It really doesn't require a lot except that you got to keep the paper out. Particles is what essentially you're looking at, and you have to keep it clean. But again, this is the other side of our quality control room. And as you can see at the back of this, there's a window. Besides this regular production area, we've actually developed an ISO 7 facility at Washington University as well. And this has been done so that we can work with industry partners. A lot of the industry partners want more than what is really required in the law. Of course, it tends to ratchet up requirements too eventually as far as what is required. But we've gone forward with this, and in the ISO 7, we actually do wear the bunny suits. We do wear masks, face covers. I'm the model here, and this is my engineer, my co-director. And again, what you're doing essentially is going into your airlock area. So again, all the air from the ISO 7 comes out to the lower grade air. So again, when you walk into this airlock, then you close the door and you open the door and enter the ISO 7 facility. And in there we have a bank of cells, hot cells for doing fluorine-18 work. We have a bank for doing carbon-11. There are 19 hot cells in this area. And this is a biosafety cabinet where we make some of our copper-64, like their kit formulation types, similar to what your technetium would be. And again, we have these airlock windows. We pass our quality control sample from the ISO 7 into our QC area as well. We also do shipping, and so this goes out into our hallway. But these are airlocks, just like the entry into the facility for the person. Anything that goes out has to go out through airlocks. So now a bit about pharmaceuticals that we are currently using and on the market, and also some that are experimental at this time. So the first one is N13 ammonia. N13 has a 10-minute half-life. We actually hold an ANDA for this product. Again, we produce it, O16, bombarding with protons to give us the N13. And essentially, it's an FDA-approved drug. It is cyclotron produced, and complex uptake mechanisms, essentially diffusion of the uncharged lipophilic ammonia. Then it's fixed as glutamine by enzymatic conversion of the glutamic acid. And this product then is actually excreted through the kidneys. Not a lot of institutions are using this. They're also using the rubidium, which is approved generator, rubidium-82. And as far as the actual process, we just take the O16, bombard it with the protons, and we form the ammonia. Problem is that ammonia is very unstable in water, so we actually have to add a 5 millimolar solution of ethanol, essentially as a scavenger, so that we really end up with ammonia as our final product. Otherwise, we get nitrites or nitrates formed. So it's really simple. We just take an IV bag, we add ethanol into the bag, shake it up, and then that's actually what we go in and load into our target to bombard to produce the N13. And this is just at Washington University. We've been fortunate we have a PET-MR, so we haven't yet been able to start using this for clinical studies. We're using it for research studies, but we have a kind of a conflict between our hospital and the research group. The research group owns the MR, PET-MR, and so they really haven't liked that approach. You know, it's kind of negotiating as to who gets what in terms of payment for the studies. We did have to install safety doors on the research area so that we could use this for clinical patients. All of that has been accomplished, and hopefully we will eventually get to use this. The nice thing for us is, as you can imagine, 10-minute half-life. We're spread out across the campus. You know, a lot of newer places. Our first medically used cyclotron was installed at Washington University in the 60s. So part of our operation is still in the basement of one hospital. The newer machines are over 2 blocks away from that. And our imaging suites are up on the 10th floor of the building where we have our older cyclotrons. So we have to travel these materials to the site. For fluorine, it's easy. You know, we can manually cart our product over to the clinic. But for N13, we can cart it to the clinic. We make about 150, 170 millicuries. So if we essentially finalize it, put it in our shipping cart, send it over to the clinic, it takes us 10 to 15 minutes to get there. So we've already lost over a half-life by the time we get there. So that means you need patients ready to be injected. It's really a lot of fun. Whereas we also have a pneumatic line that's, you know, like bank transfer systems. They're plastic. And actually, we've built tungsten shields for syringe shields and for vial shields. So we can use those in the plastic containers. But the impact, when those little, you know, they weigh about 6-7 pounds with the lead in it. And when they're delivering over, it's 2,000 feet from our cyclotron to our imaging site. It only takes a couple minutes to get there, and then they just smash down. So we've had to put essentially some, like, I can't think of the word, I always forget this word, but it's like cushion, you know, we actually have springs in there that provide resistance to that, so the plastic cases don't break. Anyway, what you're seeing here, this is a patient. It was a questionable stress rest, maybe technetium study, and a larger person. So they came in again, and there is reduced perfusion on the stress image. And also what you can see, we're using contrast agent as well. So here's the PET-MR and contrast fused agent. So we're happy, and hopefully someday we'll actually get to use it. We do use it for a lot of other studies, but we just haven't moved our ammonia forward. This is an old image that I had, but it's kind of interesting in terms of looking at viable myocardium. This is essentially the combination of using ammonia and FDG. And you can see in this top image, again, significant reduced perfusion, no perfusion, actually both on stress and in rest. And then we injected the FDG, and again, similarly, there was a match defect. So again, we know that we had non-viable myocardium. And with this one, again, when we, same thing, perfusion defect, but then we have the FDG where there's actually uptake into the myocardium. So we know we're just looking at hibernating myocardium. So again, we have recently just done a few of these studies, but not up on our PET-MR. Also, investigational-wise, there is, if you think about it, in terms of commercialization, really the only compounds that can be commercialized are something that has a bit longer half-life. Or otherwise, you could make it for your own institution, like C11 choline, but you can't really distribute a 20-minute half-life. So right now, Lantheus is actually trying, and they are in third phase development of fluridipaz. This is a perfusion imaging agent. And I just spoke actually with the Lantheus rep yesterday, or Saturday, and they said they're waiting for the final approval to move forward on their phase 3 trial. So hopefully this is coming along and we'll actually see this because it would be a great agent to have. And again, this is just compared to the SPECT-MIBI agent. So I'm going to talk a little bit about neuroimaging agents. We have a number of them that are approved on the market. We actually are not making those agents at our institution. There is an institution close to us. Avid Pharmaceutical has contracted, well, each of these companies, GE, Avid, and Piramal, have contracted sites in the United States, so centralized nuclear pharmacies, PET nuclear pharmacies, to produce these agents for them. And again, this is just a bit of history. This is the PIB, C11 PIB, the amyloid agent that's used for Alzheimer's imaging that was developed by Klunk and Mathis in Pittsburgh in 2004. This is a thioflavin dye that, again, is an amyloid binding dye. And so, again, they developed this agent, the C11 PIB, to simulate the thioflavin. And again, as I mentioned, there are F18 tracers for this amyloid that have now been developed. But in essence, in the normal, you just see essentially after injection, it's quickly washed out of the brain, and in the Alzheimer's patient, it's retained. Okay, next. Okay, so what we're looking at is the comparative is just the MR to the C11 PET and PIB, and comparing the normal versus the Alzheimer's patient. Again, not much difference in the MR scan, but there is certainly difference in the PET image. These are the 3 agents. I won't really discuss them to great detail, but they all look very much the same. And they are approved essentially for the same thing. So Avid, Vizamyl, and actually it's Amyvid, Vizamyl, and Neurosec are the 3 approved agents. The one real difficult thing for the PET industry as a whole was when these 3 agents came on the market, CMS won't reimburse for them. I mean, it took them a while to get around to that, but they essentially have a non-payment decision on these agents. So after these companies have spent a lot of time, it's been very difficult to move these agents forward and to keep new, essentially, agents coming forth from R&D. So right now, they've opened up an IDEAS study that will allow us to enter patients that CMS will cover for 3 years. And there's criteria, and you can use any one of these 3 agents to essentially look at potentially diagnosing Alzheimer's. This is just the Amyvid, and in the first upper left, you see a cognitively normal patient. And then here you have essentially a full amyloid AB positive amyloid disease. This is actually part of a study, began as a mild cognitively impaired patient, and then through the course of the study, which was a couple of years, progressed to full Alzheimer's. These are just Vizamyl. Again, images look similar, same indication for the use of these agents. And this is the florbetapen, the Neurosec from Piramal. One other area in neuroimaging that I wanted to talk about today is F18 fluorodopa. It's Parkinson's disease. We do a lot of this at Washington University. There actually is a fluorodopa application at the FDA. North Shore in New York State had their application with the FDA for significant time, I think maybe several years now. And nothing is moving forward, so we really don't know why. I actually would be very happy if we could get this out in an approved status, kind of thinking about talking with, well, I have talked with Paula Jacobs at NIH to see if potentially they might be interested in filing the NDA, and then that allows us to just submit the generic drug application. So again, we're looking at progressive degenerative disease, loss of nigrostriatal dopaminergic system, which underlies major motor manifestations of PD. Symptoms emerge when you essentially see 80 to 90 depletion of these neurons. So again, just to take a look at a couple of these. In essence, what you're imaging essentially is the area in the substantia nigra. There's a decrease in the dopaminergic neuron, which is located in the basal ganglia, and is specifically in the substantia nigra. Fluorodopa does selectively localize in these neurons. And again, as Parkinson's disease progresses, it correlates very well with the severity of PD. This is just a schematic of how the fluorodopa is taken up into the neuron. So again, it comes in as L-fluorodopa, F18, and it travels across into the neuron, where it again is transferred by enzymatic process to dopamine. So it becomes fluorodopamine. Then it's actually maintained in the vesicles until there's a essential release from the neuron, and this is the this is presynaptic neuron, postsynaptic neuron, and actually then the fluorodopa travels across the space and binds at the postsynaptic cell. So in essence, you're actually visualizing what's ongoing right at the neuronal site. This is just an example from normal to mild to moderate Parkinson's disease, and you can see, again, the way, and this is the typical progression of this disease. And again, you'll see on the right kind of is maintained. This one essentially degenerates typically a bit more rapidly. Also, fluorodopa has been used in glioma imaging, which is pretty exciting. It's a most common form of primary brain tumor, difficult to diagnose. It's diagnosed with MRCT and PET. Fluorodopa PET is useful in the detection of recurrent gliomas. Also, optic gliomas. We've done some studies with this in kids at Washington University. Rare, occurs in children usually before the age of 20, and can affect one or both optic nerves, and again, essentially the optic chiasm, which is excuse me. Maybe by the time I'm done, I'll get it right. Okay, the chiasm. So you can see a localization either in the optic area or in the chiasm. So again, what's happening in terms of its uptake into the glioma is that it's amino acid transporter. The LAT1 is, you'll find in my continuing slides that it's responsible for membrane transport of large neutral amino acids in normal cells. So this is actually a mechanism for a number of other drugs as well. And again, the study assessed the relationship between LAT1 and fluorodopa uptake in human astrocytomas, and they found that the LAT1 mRNA and protein expression varies in GBM, and the extent of the fluorodopa uptake was positively correlated with the endogenous LAT expression. So again, this work was done in, I think, 13, sorry. And this is just an example of fluorodopa detection of glioma compared to FDG, and you can see the FDG scan here. And the sensitivity for FDG is about 61% versus the fluorodopa, there's about 98%. So again, you've got significant difference in the two radiopharmaceuticals. And this again is the pediatric case I was talking about with the optic pathways glioma. And again, you can see the uptake in the optical area, the optic area. Well, in prostate cancer, this is an area that's really grown in terms of interest. Certainly, I want to give you a little bit of history. Acetate is an agent we used at Washington University for years. Some people are still actually using it, but the C11 choline now has been approved. Mayo Clinic submitted it to the FDA, and they were able to get that product approved. We have a pending ANDA at the FDA, and hopefully we've been told we should hear something by June. So very exciting. There's only other two places but Mayo in the US that have filed and gotten approved as Zevacor, which is a centralized nuclear pharmacy, but they're affiliated with a hospital in Indianapolis, and they also, one of the hospitals in Texas. I'm thinking it might have been MD Anderson, but I'm not sure. But they got the approval for choline. So with this, just a bit of information looking at estimated new cases for prostate. Again, this is from 2014, about 233,000 in males, and also in terms of estimated deaths, about 23,000, 29,000. So it is the most common malignancy in men. Lifetime risk of 1 in 6 will acquire the disease. Greater than 20% of men will die from their disease. So there's a real interest in terms of being able to diagnose this disease. C11 acetate, as I mentioned, again, the comparative, this is just pre-post, this is normal biodistribution. So you see activity normally in the pancreas as well as in the liver. And again, this is the posterior. And with the F18 FDG, again, you have the clearance organs. You know, you're seeing the ureters and the bladder, which if you have metastatic disease in the abdomen, it's going to be really difficult to try and differentiate the two. So that's why it hasn't been too useful in terms of prostate diagnosis. This is just, again, C11 acetate patient, again, recurrent comparative to FDG, see the ureter, whereas in the acetate PET, you're able to actually visualize the nodes. So C11 choline was very exciting news when this happened. It's intended for biochemical recurrence of prostate cancer. FDA got their approval in 2012. Choline is transported and metabolized. Metabolism is increased in prostate cancer. Essentially, it's a marker of cell membrane synthesis. Similarly, with acetate, it's used in cell membrane synthesis. So the clinical volume was even high. By 2014, 150 patients a month. That's a huge number of subjects. And also, they've been able to get local CMS provider-approved reimbursement, and that began in 2013. So that's another key piece for using any of these PET drugs, because we have real problem with reimbursement. So this has gone forward. The group Zevacor in Indianapolis has just gotten their pass-through, so they're able to charge for them as well. So we're hoping in June that we will be able to do the same thing at WashU. This is just an example. It's courtesy of Mayo Clinic. Again, you're seeing normal liver and spleen, but again, in the abdomen, you're seeing clear, and you can see the metastatic disease. And another example, transaxial slices, and you can see these are PET-CT images correlating to the uptake. A new agent that's actually on the market actually has now been submitted to the FDA, Blue Earth Diagnostic. This actually, the compound is FACBC, or flucyclobutane, sorry. But this was a drug that GE actually developed, patented. They didn't develop it. They took it, essentially they purchased the patent rights from the person who developed the compound. But they never ever went forward to market the drug. And so recently they spun it off, and Blue Earth, a company that's in the UK, has taken up and brought this to market now. And a number of things about this particular submission are exciting to me, because it's just difficult to work with the FDA. Though they have become more receptive, more willing to deal with the community. There was a meeting at Hopkins last March, not this year, last year, with gallium tracers, and there was an FDA, Lou Marzallo was there, he's head of imaging at the FDA, and there were also patient advocates in the audience. And the patient advocates stood up and said, why do we have to go to Europe to be able to do, the diagnosis and therapy, which was gallium agents that they were referring to, and lutetium for the therapy. And the FDA, Lou said, bring me an application and we'll move it forward rapidly. So actually that's also happened, so I'll tell you. But this one again went in to FDA, and again, this is an artificial amino acid analog, and it's labeled with fluorine 18. So again, this makes it possible in terms of distribution of this compound. And the preclinical studies do show preferential uptake into cells and tissues with enhanced amino acid transport, kind of the same ongoing theme that we're seeing. Prostate, like other tumors, has increased needs for amino acids, inducing overexpression of amino acid transporters. So again, I think this is a very exciting first step, or huge step. And here are the actual images. This is normal biodistribution. You can see there's liver and pancreas, similar to the choline, but it is a bit clearer in the abdominal area than the choline. So again, we never really know, which of these agents will be the agent, or if it will require more than one agent to actually diagnose the full extent of the disease. But again, I think it's very exciting to think about this. Also, they've looked at essentially the normal uptake. There is also salivary gland uptake. Muscle increases with time, and marrow, which is good, decreases with time. So again, and it's cleared through the bowel, absent through the kidneys, hopefully. Okay, and this is just, again, a recent update on, as far as what they have actually done. They've submitted this NDA application to the FDA. And this review, this is something that the FDA hasn't been willing to do up until now. This is not just US clinical trials. This involves individuals from a number of countries. So again, US, Norway, and Italy. So they've allowed all that data to go forward. SNMMI has been involved in working with the group in Italy, kind of, again, trying to come up with similar kinds of data collection systems, because that's a real part that's difficult with clinical trials. You need your scanners, essentially all looking at the same thing, all collecting the same data in the same formats. So again, it's a very important piece of this puzzle. Locate trial they're in phase 3. They're going to be opening 20 sites in 2016 and determine the effect of the fluocinolone imaging on management of patients with biochemically recurrent prostate cancer. This is just a comparative that was done. This study was done in Italy. And again, what they found was that FACBC was able to detect twice as many lesions as choline. This was a small patient study, so it may not have a lot of relevance. But again, they're seeing very positive results. And again, just another FACBC transaxial image, and you can see the fused. This looks like PET MR to me. This one came from Emory. And the other thing that's not a PET agent, but I wanted to mention this today, this is something that's kind of energized nuclear medicine, is therapy. We've had a couple of agents that have been used for treating lymphomas, antibody-based agents, and they've been difficult in terms of the sale to the public. It's just the utilization has really not worked very well. There was an I-131 antibody, Bexar, that's been removed from the market already, and there still is one remaining, but again, the utilization is low. So with this agent, again, radium in the periodic chart sits with calcium, strontium, magnesium. And essentially what this agent is the bone seeking. You don't need to attach it to another chelate or ligand to deliver it to the tumor. Essentially, if you're looking at metastatic bone disease, the radium, essentially the radium chloride will be delivered directly to the bone. It's being dosed by package insert at about 1.35 microcurie per kilogram, and it's a 6 injection series. So again, I know that now that it is approved, they've upped the dose. I believe they're doing clinical trials at about 2.7, I think it is, microcuries per kilogram. They're doing clinical trials in women as well, looking at bone lesions from breast cancer. And the last group of compounds are kind of a few different agents: gallium dotatok, then the gallium lutetium PSMA, and zirconium herceptin. Obviously, all are in research mode. Gallium generator, it's like the technetium generator in some ways, and in others it's not. Essentially, it provides gallium-68. It's eluted, but the problem with that generator is it's eluted in hydrochloric acid, so you can't just inject that final product. It has to be essentially purified and then added to possibly a kit formulation. So it's not like technetium in that the FDA has already stated they will not approve this generator as an approved generator. It will essentially, currently, dotatate has gone into the FDA as well by AAA. This is another positive, the dotatate. It will be labeled with gallium, and again, it's a kit formulation. then you elute the generator, essentially add it through a sterile filter into the final product vial. And that gallium then, when it's at the FDA, they're referencing Eckert Ziegler, manufacturer of the gallium generator. So that initially that's the only generator that is approved, but they are moving forward towards adding other vendors so that you would be able to use those other vendors in terms of producing the gallium dotatate. The actual parent of the generator is germanium, and this is produced in the cyclotron as well. Zinc-66 bombarded with alphas, the alpha-2N to give you the germanium, or germanium-69 to do the P2N reaction and get the gallium. And then again, this generator, this product, again, the source of the germanium is another problem. It's actually currently made at Brookhaven National Laboratory, so it's not in the typical commercialization field. But the DOE makes isotopes for human use. They also make strontium for the strontium rubidium generator, but they're always looking for partners, essentially commercial groups that will take this on and provide the isotopes for the nation, and then they will back out of the production. But currently, the germanium is being produced at Brookhaven. Zevacor, this company that I've mentioned, they've just are in the process of building 70 MeV cyclotron in Wisconsin. So it's a very large capacity. They will be able to be producing germanium and rubidium, the strontium for the two generators, one that is approved and one that hopefully won't be approved, but it will be approved with the use of the gallium dotatate. And again, long life parent. This generator is good for maybe 6 to 8 months. It depends on the length. And then you have to look at the half-life of gallium. It's 60 minutes, so 68 minutes. So again, if you think about distribution, and AAA is actually considering distribution to about maybe a 90-minute driving radius from the place where it's essentially. The reason they're thinking about this is because the cost of the generator is about $60,000, and then there will be the price of the kit formulation, which I would guess it's certainly to be at least $3,000 a kit. So again, it's a significant cost to have that in-house, and these are for neuroendocrine imaging, the dotatate. Again, there's not a large number of people that'll utilize the product. So again, you kind of have to be have a business plan that will allow you to essentially cover the United States. This is the look at the generator. This is the actual gallium generator, and this is just the labeling system. So you first initially elute the generator, and it's transferred over then to the labeling, where again you're going to purify the gallium, and then you will be adding it. You can do a more complicated type of labeling with this system, or if we ending up buying those kit formulations of the dotatate, then we just need to elute the generator, purify the gallium, and then add it to the final product, like with the technetium kit formulations. So just an example, the current agent used for on the market is Octreoscan. It's an indium labeled product that looks at these neuroendocrine tumors. And again, it's kind of a messy looking slide, or image, excuse me. And here's the gallium dotatate. But what you can say certainly is that there's significant more disease noted with the gallium dotatate than there is with the Octreoscan. This is another example where, again, a primary tumor, a patient returned, there were problems, and again, there were no determination made with the Octreoscan, but with the gallium dotatate, they were actually able to determine where the tumor was located. So it does have orphan drug status, which again is good. It does allow use by that particular population. So again, it's a small population of people if you're talking orphan drug status. And gallium dotatate, again, for meningioma uptake. So you're looking at PET-CT here, and then we get on to the PET-MR. And with the PET-MR, actually both PET-CT and PET-MR were able to determine not only the primary, but there was a secondary focus of activity on the fused images that you can see. So there is hope that these agents will come, and potentially the next step will be therapy. Because in Europe, what they're using is the gallium agent for diagnosis, and then using the same ligand attached lutetium And Zevacor does have plans as well to produce the the lutetium for use in therapeutic applications. So again, you're just looking at the uptake in prostate cancer with gallium PSMA. So again, a different ligand, different product, still a kit formulation, and the gallium generator. So the hope is that we will have a number of these kit formulations coming forward for use in the very near future. And that's just the PSMA PET image and the CT. Last topic for me is, and I know I've got just a few minutes, Immuno PET, radioimmuno PET. And again, antibodies, again, these are the agents that can direct the radioactivity to the spot of antibody localization. Antibodies are very selective targeting agents. A wide number of antibodies have been developed as therapeutic agents in the last 2 decades. So as far as Immuno PET, that means the labeled antibody, it offers the potential to stratify patients that may benefit from the antibody therapy and monitor the course of therapy, pave the way for next generation targeted radiotherapies. So again, this is certainly in the investigational stage. But similarly, FDG is used to track the process of routine chemotherapy, pre-post administration. So again, it's good to be able to gauge if an agent does work rather than continue with an expensive agent that isn't benefiting the patient and may have significant side effects. Trastuzumab, which is Herceptin, again, it's an FDA-approved antibody for the treatment of metastatic breast cancer overexpressing HER2. And Herceptin imaging, again, may be useful for predicting response to Herceptin therapy and determining the dosing strategies. This is actually an ongoing clinical trial at Washington University with zirconium-labeled trastuzumab. Zirconium has a 3.3 roughly half-life. And so why is that a good thing to be using with antibodies? Intact antibodies take quite a while to clear out of your blood into the target tissue. So you have to have a nuclide that's half-life is comparable to the biological clearance so that you can have the antibody injected, taken up into the tissue, and be available still radioactively labeled for imaging. So again, this is a good combination, and this is another, as I mentioned, nuclide we're making at Washington University. Again, useful for pharmacokinetic studies of the intact antibodies. And again, the production of the zirconium is yttrium-89 parent proton in, neutron out, PN reaction to make the zirconium-89. And also, like I mentioned, the radium goes directly to the bone, so you don't have to worry about complexing it. But with antibodies, they don't. They target the tissue, but they won't take the radioactivity there. So we have to add typically a bifunctional chelate. We add one arm of the chelate to the antibody, and then the other arm will complex the metal, the radiometal. So we first have to, stepwise, take the antibody, attach the chelate, purify excess chelate or ligand, and then add the radioactivity to the purified conjugate. And this has been used, again, to evaluate HER2-positive lesions and looked at the safety for the patient and performed human dosimetry and safety evaluation. And this is just a same study. This is an axial slice looking at the antibody in a patient with metastatic HER2-positive disease and a known femur lesion. So we've actually been able to see the lesion on the PET-CT, sorry, PET-CT. Not positive. You all know that answer. So anyway, this is kind of where we are headed. Hopefully some of these things will actually come to fruition. This is just another example where a vertical metastasis seen on MRI was unapproachable for biopsy. So the HER2 status was determined by zirconium-89 image. So again, you can see the uptake in the spine. And this is a HER2-positive brain lesion, which was undetected by conventional CT. A lesion was identified using the antibody, the zirconium antibody, and was subsequently then confirmed by the MR. So, and that's my magic ball, my crystal ball. And probably what I would say is the things moving forward will be the gallium agents, both for neuroendocrine tumor and for prostate imaging, PSMA, and hopefully, that will give us a new scope of of radiopharmaceuticals and something, again, to energize the field. So thank you very much. If you have any questions, I'll try.